of its cilia, like an independent organism, but it has no cell membrane. Similar facts were discovered through the study of the animal cell, which could not be reconciled with the old conception of the cell. A few years after the enunciation of Schwann's theory, various investigators, Kolliker, Bischoff, and others, observed many animal cells in which no distinct membrane could be discovered, and in consequence, a lengthy dispute arose as to whether these bodies were really without membranes, and hence not cells, or whether they were true cells. Further, movements similar to those seen in plant protoplasm were discovered in the granular ground substance of certain animal cells, such as the lymph corpuscles. In consequence, Remak applied the term protoplasm, which Moll had already made use of for plant cells, to the ground substance of animal cells. Important insight into the nature of protoplasm was afforded by the study of the lowest organism, Rhizopoda, or amoeba, Myxomycetes, etc. Dujardin had called the slimy, granular, contractile substance of which they are composed, sarcodae. Subsequently, Max Schultze and de Bary proved, after most careful investigation, that the protoplasm of plants and animals and the sarcoda of the lowest organism are identical. In consequence of these discoveries, investigators such as Nageli, Alexander Braun, Leidig, Kolliker, Kahn, de Bary, etc., considered the cell membrane to be of but minor importance in comparison to its contents. However, the credit is due to Max Schultze, above all others, of having made use of these later discoveries in subjecting the cell theory of Schleiden and Schwann to a searching, critical examination, and of founding a protoplasmic theory. He attacked the former articles of belief, which it was necessary to renounce, in 4 excellent, though short papers, the first of which was published in the year 1860. He based his theory that the cell membrane is not an essential part of the elementary organisms of plants and animals on the following 3 facts. First, that a certain substance, the protoplasm of plants and animals, and the sarcoda of the simplest forms, which may be recognized by its peculiar phenomena of movement, is found in all organisms. Secondly, that although, as a rule, the protoplasm of plants is surrounded by a special firm membrane, yet under certain conditions, it is able to become divested of this membrane and to swim about in water, as in the case of naked swarm spores. And finally, that animal cells and the lowest unicellular organisms very frequently possess no cell membrane, but appear as naked protoplasm and naked sarcoda. It is true that he retains the term cell, which was introduced into anatomical language by Schleiden and Schwann, but he defines it as a small mass of protoplasm endowed with the attributes of life. Hence it is evident that the term cell is incorrect. That it, nevertheless, has been retained, may be partly ascribed to a kind of loyalty to the vigorous combatants, who, as Brücke expresses it, conquered the whole field of histology under the banner of the cell theory, and partly to the circumstance that the discoveries which brought about the new reform were only made by degrees, and were only generally accepted at a time when, in consequence of its having been used for several decades of years, the word cell had taken firm root in the literature of the subject. End of section 7. Section 8 of the Science, History of the Universe, Volume 5. This is a LibriVox recording. All LibriVox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. The Science, History of the Universe, Volume 5, edited by Francis Rolt Wheeler. Biology, Chapter 5, Cell Life, Part 2. Since the time of Brücke and Max Schultze, knowledge of the true nature of the cell has increased considerably. Great insight has been gained into the structure and the vital properties of the protoplasm, and in especial, knowledge of the nucleus, and of the part it plays in cell multiplication, and in sexual reproduction, has recently made great advances. The earlier definition, the cell is a little mass of protoplasm, must now be replaced by the following. The cell is a little mass of protoplasm, which contains in its interior a specially formed portion, the nucleus. It is evident from the preceding history of the cell theory that the term cell is a biological misnomer, for cells only rarely assume the form implied by the word of hollow chambers surrounded by solid walls. The term is merely a historical survival of a word casually employed by the botanists of the 17th century to designate the cells of certain plant tissues, which, when viewed in section, give somewhat the appearance of a honeycomb. The cells of these tissues are, in fact, separated by conspicuous solid walls, which were mistaken by Schleiden, followed by Schwann, for their essential part. The living substance contained within the walls was at first overlooked or was regarded as a waste product, a view based upon the fact that in many important plant tissues, such as cork or wood, it may wholly disappear, leaving only the lifeless walls. Researches showed, however, that most living cells are not hollow but solid bodies, and that in many cases, for instance, the colorless corpuscles of blood and lymph, are naked masses of protoplasm not surrounded by definite walls. Thus it was proved that the neither vesicular form nor the presence of surrounding walls is an essential character, and that the cell contents, for example, the protoplasm, must be the seat of vital activity. Within the cell contents lies a body, usually of definite rounded form, known as the nucleus, and this in turn often contains one or more smaller bodies or nucleoli. By some of the earlier workers, the nucleus was supposed to be, like the cell wall, of secondary importance, and many forms of cells were described as being devoid of a nucleus. Cytoids of Haeckel. Nearly all later researches have indicated, however, that the characteristic nuclear material, whether forming a single body or scattered in smaller masses, is always present, and that it plays an essential part in the life of the cell. Besides the presence of the protoplasm and nucleus, no other structural features of the cell are yet known to be of universal occurrence. We may, says Wilson, therefore still accept as valid the definition given more than 30 years ago by Leidig and Max Schulze, that a cell is a mass of protoplasm containing a nucleus, to which we may add Schulze's statement that both nucleus and protoplasm arise through the division of the corresponding elements of a pre-existing cell. The form of cells is highly variable. In isolated cells, especially those floating freely in a fluid and not subjected to unequal pressure, the spherical form is common, but even such free cells may be modified in form by internal movements and differentiations of the cell substance. For example, some egg cells are spherical, while others are ovoid. Muscle cells are elongated, nerve cells much branched, and the white cells of the blood are irregular in shape because of their movements. But no matter how diverse the form of the cells, their structure is essentially the same. As already suggested, a cell wall or membrane is usually, though not always, present. Sometimes the cell substance has no more of a limiting membrane than has a drop of oil floating in water. That is, there is simply an undifferentiated film separating it from its surroundings. The cell mass or cell substance consists of protoplasm, the active living substance, in which may be embedded granules of lifeless substances. If the term protoplasm is accepted as synonymous with living matter, then there is protoplasm both in the main body of the cell and also in the specifically differentiated mass, most commonly central in position, known as the nucleus. It is convenient to call the protoplasm outside the nucleus cytoplasm, and that within the nucleus the karyoplasm or nucleoplasm. In the cytoplasm, there are various lifeless substances, metaplasm. Some of these, like fat and starch, are reserve food absorbed but not yet used by the cell. Others, like pigment and cell wall, are the lifeless products of life activity. The amount of metaplasm is frequently vastly greater than the amount of protoplasm in a cell. For example, a hen's egg just before leaving the ovary is a cell about 1 inch in diameter, the yolk of the egg. A small white disk on the upper surface consists of concentrated protoplasm, but the greater part of this enormous egg cell is made up of stored yolk, metaplasm, to be used as food by the chick in its development during incubation. The nucleus is usually surrounded by a definite membrane, the nuclear membrane. Within this membrane, and embedded in the general protoplasmic basis, there are granules or masses of a substance which has a strong affinity for certain chemical dyes, hence called chromatin. When this chromatin is massed into rod-shaped bodies, as happens in the process of cell division, the term chromosomes is applied to these masses of chromatin. Then, too, other bodies, nucleoli, are often present. Their nature is not clearly understood, probably because they are extremely variable. Careful study of the nucleus during all its phases gives reason to believe that its structural basis is similar to that of the cell body, and that during the course of cell division, when the nuclear membrane usually disappears, cytoplasm and karyoplasm come into direct continuity. For these and other reasons, the term nucleus and cell body should probably be regarded only as topographical expressions denoting two differentiated areas in a common structural basis. The terms karyoplasm and cytoplasm possess, however, a specific significance owing to the fact that there is, on the whole, a definite chemical contrast between the nuclear substance and that of the cell body, the former being characterized by the abundance of the substance rich in phosphorus, known as nuclein, while the latter contains no true nuclein and is especially rich in albuminous substances such as nucleoalbumins, albumins, globulins, and the like, which contain little or no phosphorus. Both morphologically and physiologically, says Wilson, the differentiation of the active cell substance into nucleus and cell body must be regarded as a fundamental character of the cell because of its universal or all but universal occurrence, and because there is reason to believe that it is in some manner an expression of the dual aspect of the fundamental process of metabolism, constructive and destructive, that lies at the basis of cell life. In addition to the cytoplasm or cell body and the nucleus, recent biologists believe that an extremely minute body lying just outside the nuclear membrane and known as the centrosome is an essential element of the cell. The centrosome has been seen in a large number of cells, is known to play an important part in cell division and in the fertilization of egg cells, and has been regarded by some cytologists as the dynamic center of the cell. It is still under investigation, and the interested reader should refer to such technical books as Wilson's The Cell in Development and Inheritance for the latest studies of its structure. Some animal and plant cells appear to differ so much from one another as to their form and contents that at first sight they seem to have nothing in common, and hence it seems impossible to compare them. For instance, if a cell at the growing point of a plant be taken and compared with one filled with starch granules from the tuber of a potato, or the contents of an embryo cell from a germinal disc be compared with those of a fat cell, or of one from the egg of an amphibian filled with yolk granules, the inexperienced observer sees nothing but contrasts. Nevertheless, all these exceedingly different cells are seen on closer examination to be similar in one respect, for example, in the possession of a very important, peculiar mixture of substances, which is sometimes present in large quantities, and sometimes only in traces, but which is never wholly absent in any elementary organism. In this mixture of substances, the wonderful vital phenomena may very frequently be observed, contractility, irritability, etc. And moreover, since in young cells, in lower organisms, and in the cells of growing points and germinal areas, it is in the cell substance alone, the nucleus of course being excepted, that these properties have been observed. This substance has been recognized as the chief supporter of the vital functions. It is the protoplasm or forming matter. In order to know what protoplasm is, it is advisable to examine it in those cells in which it is present in large quantities, and in which it is as free as possible from admixture with other bodies, and among such, the most suitable are those organisms from the study of which the founders of the protoplasmic theory form their conception of the nature of this substance. Such organisms are young plant cells, amoeba, and the lymph corpuscles of vertebrates. The protoplasm of unicellular organisms and of plant and animal cells appears as a viscid substance, which is almost always colorless, which will not mix with water, and which, in consequence of a certain resemblance to slimy substances, was called by Schleiden the slime of the cell. Its refractive power is greater than that of water, so that the most delicate threads of protoplasm, although colorless, may be distinguished in this medium. Minute granules, the microsomes, which look only like dots, are always present in greater or less numbers in all protoplasm, and may be seen with a low power of the microscope to be embedded in a homogeneous ground substance. As to the very minute structure of protoplasm, most of the earlier observers, says Wilson, regarded the meshwork as a fibrillar structure, either forming a continuous network or reticulum, somewhat like the fibrous network of a sponge, or consisting of disconnected threads, whether simple or branching, filar theory of Fleming. And the same view is widely held at the present time. The meshwork has received various names in accordance with this conception, among which may be mentioned reticulum, threadwork, spongioplasm, mytome, filar substance, all of which are still in use. Under this view, the granules described by Schultz, Virchow, and still earlier observers have been variously regarded as nodes of the network, optical sections of the threads, or as actual granules, microsomes, suspended in the network as described above. Widely opposed to these views is the alveolar theory of Bucheli, which has won an increasing number of adherents. Bucheli regards protoplasm as having a foam-like alveolar structure, Wabenstruktur, nearly similar to that of an emulsion, and he has shown in a series of beautiful experiments the artificial emulsions, variously prepared, may show the microscope a marvelously close resemblance to living protoplasm, and further, that drops of oil emulsion suspended in water may even exhibit amoeboid changes of form. The 2 or 3 general views, hereinbefore outlined, may justly be designated respectively as the fibrillar, reticular, or filar, and alveolar theories of protoplasmic structure, and each of them has been believed by some of its adherents to be universally applicable to all forms of protoplasm. Beside them may be placed, as a third general view, the granular theory especially associated with the name Altmann, by whom it has been most fully developed, though a number of earlier writers have held similar views. According to Altmann's view, which apart from its theoretical development, approaches in some respects that of Bucheli, protoplasm is compounded of innumerable minute granules, which alone form its essential active basis, and while fibrillar or alveolar structures may occur, these are of only secondary importance. Which of these views is correct? The present tendency of cytologists is toward the conclusion that none of them is of universal application and that all exist under certain conditions. In support of this may be cited the studies of Professor Wilson, who has been led to the conclusion that no universal formula for protoplasmic structure can be given. In that classical object, the echinoderm egg, for example, it is easy to satisfy oneself, both in the living cell and in sections, that the protoplasm has a beautiful alveolar structure, exactly as described by Bucheli in the same object. This structure is here, however, entirely of secondary origin, for its genesis can be traced step by step during the growth of the ovarian eggs through the deposit of minute drops in a homogeneous basis, which ultimately gives rise to the intraalveolar walls. In these same eggs, the astral systems formed during their subsequent division are, I believe, no less certainly fibrillar, and thus we see the protoplasm of the same cell passing successively through homogeneous, alveolar, and fibrillar phases at different periods of growth and in different conditions of physiological activity. There is good reason to regard this as typical of protoplasm in general. Buccioli's conclusions, based on researches so thorough, prolonged, and ingenious, are entitled to great weight, yet it is impossible to resist the evidence that fibrillar and granular, as well as alveolar structures, are of wide occurrence, and while each may be characteristic of certain kinds of cells or of certain physiological conditions, none is common to all forms of protoplasm. If this position is well grounded, we must admit that the attempt to find a visible protoplasmic structure, any adequate insight into its fundamental modes of physiological activity, has thus far proved fruitless. We must rather seek the source of these activities in the ultramicroscopical organization, accepting the probability that apparently homogeneous protoplasm is a complex mixture of substances, which may assume various forms of visible structure according to its mode of activity. Much discussion has been given to the question as to which of the visible elements of the protoplasm should be regarded as the living substance proper. Later discussions have shown the futility of this discussion, which is indeed largely a verbal one, turning as it does on the sense of the word living. In practice, we continually use the word living to denote various degrees of vital activity. Protoplasm derived of nuclear material has lost, wholly or in part, one of the most characteristic vital properties, namely, the power of synthetic metabolism. Yet we still speak of it as living, since it still retains for a longer or shorter period such properties as irritability and the power of coordinated movement, and in like manner, various special elements of protoplasm may be termed living in a still more restricted sense. In its fullest meaning, however, the word living implies the exstance of a group of coordinating activities more complex than those manifested by any one substance or structural element. I am, therefore, entirely in accord with the view urged by Sachs, Kolliker, Verworn, and other recent writers that life can only be properly regarded as a property of the cell system as a whole, and the separate elements of the system would, with Sachs, better be designated as active or passive, rather than as living or lifeless. Thus regarded, the distinction between protoplasmic and metaplasmic substances, while a real and necessary one, becomes after all one of degree. End of section 8. Section 9 of the Science, History of the Universe, Volume 5. This is a LibriVox recording. All LibriVox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. The Science, History of the Universe, Volume 5, edited by Francis Rolt Wheeler. Biology, Chapter 6, Cell Division. Among the vital phenomena exhibited by cells and visible through the microscope, none is so strikingly distinctive of living matter as is the process of cell division. Closely connected with it are some of the greatest problems of biology. By the continued division of an original germ cell or egg cell, all the tissue cells of a multicellular animal arise, and the germ cell itself arises in the parent body from other cells by cell division. Thus the problem is one of the central facts of development and inheritance. The rapid advance of biological research is continually adding weight to the conclusions reached years ago that every cell originates by division of some pre-existing cell. Omni cellula e cellulae. This is now regarded as one of the fundamental laws of biology, and obviously is a corollary of the biogenetic law spoken of, which states that all living matter, known to exist only in cells, originates from pre-existing living matter. How do cells originate? Was the problem which troubled the biologists of the early part of the 19th century. Schleiden and Schwann tried to answer the question, but their answer was entirely wrong. They held that cells, which they were fond of comparing to crystals, formed themselves like crystals in a mother liquor. Schwann even went further, teaching that young cells developed not only within the mother cell, as propounded by Schleiden, but also outside of it, in an organic substance, which is frequently present in animal tissues as intercellular substance, and which he called also cytoplasm. Thus Schwann taught that cells were formed spontaneously, both inside and outside of the mother cell, which would be a genuine case of spontaneous generation from formless germ substance. These were indeed grave fundamental errors, from which, however, the botanists were the first to extricate themselves. In the year 1846, a general law was formulated in consequence of the observations of Moll, Unger, and above all, Nageli. This law states that new plant cells only spring from those already present, and further, that this occurs in such a manner that the mother cell becomes broken up by dividing into two or more daughter cells. This was first observed by Moll. It was much more difficult to disprove the theory that the cells of animal tissues arise from cytoblasts, and this was especially the case in the domain of pathological anatomy, for it was thought that the formation of tumors and pus could be traced back to cytoblasts. At last, after many mistakes, more light was thrown upon the subject of the genesis of cells in the animal kingdom also, until finally the cytoblastic theory was absolutely disproved by Virchow, who originated the formula omnis cellula e cellulae. No spontaneous generation of cells occurs either in plants or animals. The many millions of cells of which, for instance, the body of a vertebrate animal is composed, have been produced by the repeated division of one cell, the ovum, in which the life of every animal commences. The older histologists were unable to discover what part the nucleus played in cell division. For many decades, two opposing theories were held, of which now one and now the other obtained temporarily the greater number of supporters. According to the one theory, which was held by most botanists, the nucleus at each division was supposed to break up and become diffused throughout the protoplasm in order to be formed anew in each daughter cell. According to the other, the nucleus was supposed to take an active part in the process of cell division, and at the commencement of it, to become elongated and constricted at a point corresponding with the plane of division which is seen later, and to divide into halves, which separate from one another and move apart. Later discoveries, 1873 to 1880, revealed the extremely interesting formations and metamorphoses which are seen in the nucleus during cell division. These investigations have all pointed to the same conclusion, that the nucleus is a permanent and most important organ of the cell, and that it evidently plays a distinct role in the cell life during division. Just as the cell is never spontaneously generated, but is produced directly by the division of another cell, so the nucleus is never freshly created, but is derived from the constituent particles of another nucleus. The formula, omnis cellula e cellulae, might be extended by adding omnis nuclei e nucleo. Since about 1876, it has been recognized that there are two widely different types of cell division. In one type, there appears to be a simple constriction of the nucleus and cell body. This is known as direct akinetic or amitotic division. The second type is vastly more complicated, as will be described. To this are now applied the terms direct division, karyokinesis, or mitosis. The terms mitosis and amitosis, referring to the presence or absence of chromosomes in the dividing cells, are preferred by most biologists. As to the occurrence of these two types, modern research has demonstrated the fact that amitosis or direct division, regarded by Remak and his immediate followers as of universal occurrence, is in reality a rare and exceptional process, and there is reason to believe, furthermore, that it is especially characteristic of highly specialized cells, incapable of long-continued multiplication, or such as are in the early stages of degeneration, for instance, in glandular epithelia and in the cells of transitory embryonic envelopes, where it is of frequent occurrence. Whether this view be well-founded or not, it is certain that in all the higher and in many of the lower forms of life, indirect division or mitosis is the typical mode of cell division. It is by mitotic division that the germ cells arise and are prepared for their union during the process of maturation, and by the same process, the oosperm segments and gives rise to the tissue cells. It occurs not only in the highest forms of plants and animals, but also in such simple forms as the rhizopods, flagellates, and diatoms. It may therefore be justly regarded as the most general expression of the eternal law of continuous development on which Virchow insisted. The phenomena which occur during the process of mitosis are very varied and very complicated. Nevertheless, they conform to certain laws which are wonderfully constant in both plants and animals. The main feature of the process consists in this, that the various substances which are present in the resting nucleus undergo a definite change of position, and the nuclear membrane, being dissolved, enter into closer union with the cytoplasmic substance. During this process, the whole mass of chromatin in the nucleus becomes transformed into fine thread-like segments, the chromosomes, the number of which remains constant for each species of plant or animal. These chromosomes are arranged in a characteristic manner on a spindle-like structure of achromatic material extending between the two centrosomes. Each chromosome then divides longitudinally into two daughter chromosomes, which for a time lie parallel with each other and are closely connected. Next, these daughter chromosomes separate into two groups, dividing themselves equally between the two groups to form the foundation of the daughter nuclei. The cell itself, meanwhile, becomes divided in such a way that each of the two cells formed by the division possesses one of the daughter nuclei. The manner in which these changes are brought about is so interesting that it seems wise to give a more detailed outline of the process. Professor Wilson in The Cell in Development and Inheritance gives a description in Essentials as follows. In the present state of knowledge, it is somewhat difficult to give a connected general account of mitosis, owing to the uncertainty that hangs over the nature and functions of the centrosome. For the purpose of the following preliminary outline, we shall take as a type mitosis, in which a distinct and persistent centrosome is present, as has been mostly clearly determined in the maturation and cleavage of various animal eggs, and in the division of the testes cells. In such cases, the process involves three parallel series of change, which affect the nucleus, the centrosome, and the cytoplasm of the cell body respectively. For descriptive purposes, it may conveniently be divided into a series of successive stages or phases, which, however, graduate into one another and are separated by no well-defined limits. These are 1. The prophases, or preparatory changes. 2. The metaphase, which involves the most essential step in the division of the nucleus. 3. The anaphases, in which the nuclear material is distributed. 4. The teleophases, in which the entire cell divides and the daughter cells are formed. 1. Prophases. As the cell prepares for division, the most conspicuous fact is a transformation of the nuclear substance involving both physical and chemical changes. The chromatin substance rapidly increases in staining power, loses its net-like arrangement, and finally gives rise to a definite number of separate, intensely staining bodies, usually rod-shaped, known as chromosomes. As a rule, this process takes place as follows. The chromatin resolves itself, little by little, into a more or less convoluted thread, known as the skein or spireme, and its substance stains far more intensely than that of the reticulum. The spireme thread is at first fine and closely convoluted, forming the close spireme. Later the thread thickens and shortens, and the convolution becomes more open. In some cases there is but a single continuous thread. In others, the thread is from its first appearance divided into a number of separate pieces or segments, forming a segmented spireme. In either case, it ultimately breaks transversely to form the chromosomes, which in most cases have the form of rods, straight or curved, though they are sometimes spherical or ovoidal, and in certain cases may be joined together in the form of rings. The staining power of the chromatin is now at a maximum. As a rule, the nuclear membrane meanwhile fades away and finally disappears, though there are some cases in which it persists more or less completely through all the phases of division. The chromosomes now lie naked in the cell, and the ground substance of the nucleus becomes continuous with the surrounding cytoplasm. The remarkable fact has now been established with high probability that every species of plant or animal has a fixed and characteristic number of chromosomes, which regularly occurs in the division of all its cells, and in all forms arising by sexual reproduction, the number is even. Thus, in some of the sharks, the number is 36. In the mouse, the salamander, the trout, the lily, 24. In the ox, guinea pig, and in man, the number is said to be 16, and the same number is characteristic of the onion. In the grasshopper, it is 12. Under certain conditions, the number of chromosomes may be less than the normal in a given species, but these variations are only apparent exceptions. The nucleoli differ in their behavior in different cases. True nucleoli or plasmosomes sooner or later disappear, and the greater number of observers agree that they do not take part in the chromosome formation. Meanwhile, more or less nearly parallel with these changes in the chromatin, a complicated structure known as the amphiaster makes its appearance in the position formerly occupied by the nucleus. This structure consists of a fibrous, spindle-shaped body, the spindle, at either pole of which is a star or aster formed of rays or astral fibers radiating into the surrounding cytoplasm, the whole strongly suggesting the arrangement of iron filings in the field of a horseshoe magnet. The center of each aster is occupied by a minute body known as the centrosome, Boveri 88, which may be surrounded by a spherical mass known as the centrosphere, Strasburger 93. As the amphiaster forms, the chromosomes group themselves in a plane passing through the equator of the spindle and thus form what is known as the equatorial plate. The entire structure resulting from the foregoing changes is known as the karyokinetic or mitotic figure. It may be described as consisting of two distinct parts, namely the chromatic figure, formed by the deeply staining chromosomes, and the achromatic figure, consisting of the spindle and asters, which in general stain but slightly. 2. Metaphase. The prophases of mitosis are, on the whole, preparatory in character. The metaphase which follows forms the initial phase of actual division. Each chromosome splits lengthwise into two exactly similar halves, which afterward diverge to opposite poles of the spindle, and here each group of daughter chromosomes finally gives rise to a daughter nucleus. In some cases, the splitting of the chromosomes cannot be seen until they have grouped themselves in the equatorial plane of the spindle, and it is only in this case that the term metaphase can be applied to the mitotic figure as a whole. In a large number of cases, however, the splitting may take place at an earlier period in the spireme stage, or even in a few cases in the reticulum of the mother nucleus. Such variations do not, however, affect the essential fact that the chromatic network is converted into a thread which, whether continuous or discontinuous, splits throughout its entire length into two exactly equivalent halves. The splitting of the chromosomes, discovered by Fleming in 1880, is the most significant and fundamental operation of cell division, for by it, as Roux first pointed out in 83, the entire substance of the chromatic network is precisely halved, and the daughter nuclei receive precisely equivalent portions of chromatin from the mother nucleus. It is very important to observe that the nuclear division always shows this exact quality, whether division of the cell body be equal or unequal. 3. Anaphases. After splitting of the chromosomes, the daughter chromosomes, arranged in two corresponding groups, diverge to opposite poles of the spindle, where they become closely crowded in a mass near the center of the aster. As they diverge, the two groups of daughter chromosomes are connected by a bundle of achromatic fibers, stretching across the interval between them and known as the interzonal fibers or connecting fibers. In the division of plant cells, and often in that of animal cells, these fibers show during this period a series of deeply staining thickenings in the equatorial plane, forming the cell plate or mid-body. 4. Telophases. In the final phases of mitosis, the entire cell is divided in two in a plane passing through the equator of the spindle, each of the daughter cells receiving a group of chromosomes, half of the spindle and one of the asters with its centrosome. Meanwhile, a daughter nucleus is reconstructed in each cell from the group of chromosomes it contains. When first formed, the daughter nuclei are of equal size. If, however, division of the cell body has been unequal, the nuclei become, in the end, correspondingly unequal, a fact which, as Conklin and others have pointed out, proves that the size of the nucleus is controlled by that of the cytoplasmic mass in which it lies. The fate of the achromatic structures varies considerably and has been accurately determined in only a few cases. As a rule, the spindle fibers disappear more or less completely, but a portion of their substance sometimes persists in a modified form. In dividing plant cells, the cell plate finally extends across the entire cell and splits into two layers, between which appears the membrane by which the daughter cells are cut apart. A nearly similar process occurs in a few animal cells, but as a rule, the cell plate is here greatly reduced and forms no membrane, the cell dividing by constriction through the equatorial plane. Such is a general description of mitosis. The variations from the type, the origin and fate of the various structures taking part in the division, their relation to each other, and the mechanisms by means of which the chromosomes divide and separate to form the new nuclei are interesting problems which are today being subjected to keen investigation in the laboratories of cytologists. But most interesting of all, perhaps, is the problem of the chromosomes in relation to inheritance. This problem will be more fully discussed in the chapter on heredity. Some of the most important studies on the physiological relations of nucleus and cytoplasm have been made with one-celled animals. Brandt in 1877 and Nussbaum in 1884 cut certain protozooms into pieces and observed that pieces containing nuclear matter quickly regenerate and produce perfect animals, while the enucleated fragments soon die. One of the most remarkable animals with this power to regenerate is the trumpet animalcule, Stentor. Gruber in 1885 found that when this animalcule was fragmented, pieces possessing a large fragment of the nucleus completely regenerated within 24 hours. If the nuclear fragment was smaller, the regeneration proceeded more slowly. If no nuclear substance were present, no regeneration took place, though the wound closed and the fragment lived for a considerable time. The only exception, but it is a very significant one, was the case of individuals in which the process of normal fission had begun. In these, a non-nucleated fragment in which the formation of a new periostome had already been initiated healed the wound and completed the formation of the peristome. Lilly, 1896, has recently found that Stentor may by shaking be broken into fragments of all sizes, and that nucleated fragments as small as 1/27th of the volume of the entire animal are still capable of complete regeneration. Non-nucleated fragments perish. Many other such experiments on one-celled animals show that life for a considerable period, perfectly normal movements, susceptibility to stimulus, and the power of taking food may continue in enucleated parts of unicellular animals. They lack, however, the power of digestion and secretion, and hence cannot continue to live as do the nucleated parts. These facts demonstrate that the nucleus plays an important part in metabolism. Experiments on plants have supported this conclusion. It will be noted that if a unicellular organism be divided into two parts, the part with the nucleus is a complete cell. a mass of protoplasm with a nucleus, while the other part possesses no longer the individuality of a cell and perishes. It is well known that all animals and plants have a definite limit of growth. From the cytological point of view, the limit of the body size appears to be correlated with the total number of cells formed rather than with their individual size. This relation has been carefully studied by Conkline, 96, in the case of the gastropod Crepidula, an animal which varies greatly in size in the mature condition, the dwarfs having in some cases not more than 1/25 the volume of the giants. The eggs are, however, of the same size in all, and their number is proportional to the size of the adult. The same is true of the tissue cells. Measurements of cells from the epidermis, the kidney, the liver, the alimentary epithelium, and other tissues show that they are on the whole as large in the dwarfs as in the giants. The body size, therefore, depends on the total number of cells rather than on their size, individually considered, and the same appears to be the case in plants. It is in the cells of both plant and animal organisms that the vital functions are carried on. All the vital processes of a complex animal appear to be nothing but the highly developed result of the individual vital processes of its innumerable, variously functioning cells. The study of the processes of digestion, of the changes in muscle and nerve cells, leads finally to the examination of the functions of gland, muscle, ganglion, and brain. End of section 9.
Section 10 of The Science History of the Universe, Volume 5. This is a LibriVox recording. All LibriVox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. The Science History of the Universe, Volume 5, edited by Francis Rolt Wheeler. Biology, Chapter 7, Organic Functions, Part 1. The facts dealing with the physiology of organisms, the activities associated with that which we call life, are often designated organic functions. The terms animal physiology, plant physiology, and human physiology are in common use and often suggest to the lay reader that the functions or workings of the organs of plants, animals, or man are quite distinct, so much so as to require discussion in different treatises. This is true only as a matter of detail, for in the past 50 years it has been made evident that in general principles all living things are fundamentally similar. One of the most important summaries of this similarity is Huxley's famous essay, The Border Territory Between the Animal and Vegetable Kingdoms, written in 1876, extracts from which follow. In the second edition of the Reine Animale, published in 1828, Cuvier devotes a special section to the division of organized beings into animals and vegetables, in which the question is treated with that comprehensiveness of knowledge and clear critical judgment which characterize his writings and justify biologists in regarding them as representative expressions of the most extensive, if not the profoundest, knowledge of his time. He affirms that living beings have been subdivided from the earliest times into animated beings, which possess sense and motion, and inanimated beings which are devoid of these functions and simply vegetable. Although the roots of plants direct themselves toward moisture and their leaves toward air and light, although the parts of some plants exhibit oscillating movements without any perceptible cause, and the leaves of others retract when touched, yet none of these movements justify the ascription to plants of perception of will. From the mobility of animals, Cuvier, with his characteristic partiality for teleological reasoning, reduces the necessity of the existence in them of an alimentary cavity or reservoir of food, whence their nutrition may be drawn by vessels, which are a sort of internal roots, and in the presence of this alimentary cavity, he naturally sees the primary and the most important distinction between animals and plants. Following out his teleological argument, Cuvier remarks that the organization of this cavity and its appurtenances must needs vary according to the nature of the aliment and the operations which it has to undergo before it can be converted into substances fitted for absorption, while the atmosphere and the earth supply plants with juices ready prepared and which can be absorbed immediately. As the animal body required to be independent of heat and of the atmosphere, there were no means by which the motion of its fluids could be produced by internal causes. Hence arose the second great distinctive character of animals, or the circulatory system, which is less important than the digestive, since it was unnecessary and therefore is absent in the more simple animals. Animals further needed muscles for locomotion and nerves for sensibility. Hence, says Cuvier, it was necessary that the chemical composition of the animal body should be more complicated than that of the plant, and it is so inasmuch as an additional substance, nitrogen, enters into it as an essential element, while in plants, nitrogen is only accidentally joined with the three other fundamental constituents of organic beings: carbon, hydrogen, and oxygen. Indeed, he afterward affirms that nitrogen is peculiar to animals, and herein he places the third distinction between the animal and the plant. The soil and the atmosphere supply plants with water composed of hydrogen and oxygen and carbonic acid containing carbon and oxygen. They retain the hydrogen and the carbon, exhale the superfluous oxygen, and absorb little or no nitrogen. The essential character of vegetable life is the exhalation of oxygen, which is effected through the agency of light. Animals, on the contrary, derive their nourishment either directly or indirectly from plants. They get rid of the superfluous hydrogen and carbon and accumulate nitrogen. The relations of plants and animals to the atmosphere are therefore inverse. The plant withdraws water and carbonic acid from the atmosphere. The animal contributes both to it. Respiration, that is, the absorption of oxygen and the exhalation of carbonic acid, is the specially animal function of animals and constitutes their fourth distinctive character. Thus wrote Cuvier in 1828. But in the fourth and fifth decades of this century, the greatest and most rapid revolution which biological science has ever undergone was effected by the application of the modern microscope to the investigation of organic structure, by the introduction of exact and easily manageable methods of conducting the chemical analysis of organic compounds, and finally, by the employment of instruments of precision for the measurement of the physical forces which are at work in the living economy. That the semi-fluid contents, which we now term protoplasm, of the cells of certain plants, such as the charae, are in constant and regular motion, was made out by Bonaventura Corti a century ago. But the fact, important as it was, fell into oblivion and had to be rediscovered by Treviranus in 1807. Robert Brown noted the more complex motions of the protoplasm in the cells Atradescantia in 1831, and now such movements of the living substance of plants are well known to be some of the most widely prevalent phenomena of vegetable life. Agard and other of the botanists of Cuvier's generation, who occupied themselves with the lower plants, had observed that, under particular circumstances, the contents of the cells of certain water weeds were set free and moved about with considerable velocity, and with all the appearances of spontaneity as locomotive bodies, which, from their similarity to animals of simple organization, were called zoospores. Even as late as 1845, however, a botanist of Schleiden's eminence dealt very skeptically with these statements, and his skepticism was the more justified since Ehrenberg, in his elaborate and comprehensive work on the infusoria, had declared the greater number of what are now recognized as locomotive plants to be animals. At the present day, writes Huxley, innumerable plants and free plant cells are known to pass the whole or part of their lives in an actively locomotive condition, in no wise distinguishable from that of one of the simpler animals, and, while in this condition their movements are, to all appearances, as spontaneous, as much the product of volition as those of such animals. Hence the teleological argument for Cuvier's first diagnostic character, the presence in animals of an alimentary cavity or internal pocket in which they can carry about their nutriment, has broken down so far at least as his mode of stating it goes, and, with the advance of microscopic anatomy, the universality of the fact itself among animals has ceased to be predicable. Many animals of even complex structure which live parasitically within others are wholly devoid of an alimentary cavity. Their food is provided for them, not only ready cooked, but ready digested, and the alimentary canal becomes superfluous, has disappeared, and again, the males of most rotifers have no digestive apparatus. Finally, amid the lowest forms of animal life, the speck of gelatinous protoplasm, which constitutes the whole body, has no permanent digestive cavity or mouth, but takes in its food anywhere and digests, so to speak, all over its body. But, although Cuvier's leading diagnosis of the animal from the plant will not stand a strict test, it remains one of the most constant of the distinctive characters of animals. And, if we substitute for the possession of an alimentary cavity the power of taking solid nutriment into the body and there digesting it, the definition so changed will cover all animals except certain parasites, and the few and exceptional cases of non-parasitic animals which do not feed at all. On the other hand, the definition thus amended will exclude all ordinary vegetable organisms. Cuvier himself practically gives up his second distinctive mark when he admits that it is wanting in the simpler animals. The third distinction is based on a completely erroneous conception of the chemical differences and resemblances between the constituents of animal and vegetable organisms, for which Cuvier is not responsible, as it was current among contemporary chemists. It is now established that nitrogen is as essential a constituent of vegetable as of animal living matter, and that the latter is, chemically speaking, just as complicated as the former. Starchy substances, cellulose and sugar, once supposed to be exclusively confined to plants, are now known to be regular and normal products of animals. Amylaceous and saccharine substances are largely manufactured even by the highest animals. Cellulose is widespread as a constituent of the skeletons of the lower animals, and it is probable that amyloid substances are universally present in the animal organism, though not in the precise form of starch. Moreover, although it remains true that there is an inverse relation between the green plant in sunshine and the animal, insofar as under these circumstances the green plant decomposes carbonic acid and exhales oxygen while the animal absorbs oxygen and exhales carbonic acid, yet the exact researches of the modern chemical investigators of the physiological processes of plants have clearly demonstrated the fallacy of attempting to draw any general distinction between animals and vegetables on this ground. In fact, the difference vanishes with the sunshine, even in the case of the green plant, which in the dark absorbs oxygen and gives out carbonic acid like any animal. On the other hand, those plants, such as the fungi, which contain no chlorophyll and are not green, are always, so far as respiration is concerned, in the exact position of animals. They absorb oxygen and give out carbonic acid. Thus, by the progress of knowledge, Cuvier's fourth distinction between the animal and the plant has been as completely invalidated as the third and second, and even the first can be retained only in a modified form and subject to exceptions. But has the advance of biology simply tended to break down old distinctions without establishing new ones? With a qualification, to be considered presently, the answer to this question is undoubtedly in the affirmative. The famous researches of Schwann and Schleiden in 1837 and the following years founded the modern science of histology, or that branch of anatomy which deals with the ultimate visible structure of organisms as revealed by the microscope, and from that day to this, the rapid improvement of methods of investigation and the energy of a host of accurate observers have given greater and greater breadth and firmness to Schwann's great generalization that a fundamental unity of structure obtains in animals and plants, and that however diverse may be the fabrics or tissues of which their bodies are composed, all these varied structures result from the metamorphosis of morphological units, termed cells in a more general sense than that in which the word cells was at first employed, which are not only similar in animals and in plants respectively, but present a close resemblance when those of animals and those of plants are compared together. The contractility, which is the fundamental condition of locomotion, continues Huxley, has not only been discovered to exist far more widely among plants than was formerly imagined, but in the plants the act of contraction has been found to be accompanied, as Dr. Burdon Sanderson's interesting investigations have shown, by a disturbance of the electrical state of the contractile substance comparable to that which was found by Dubois-Reymond to be a concomitant of the activity of ordinary muscle in animals. Again, I know of no test by which the reaction of the leaves of the sundew and of other plants to stimuli, so fully and carefully studied by Mr. Darwin, can be distinguished from those acts of contraction following upon stimuli which are called reflex in animals. On each lobe of the bilobed leaf of Venus flytrap are 3 delicate filaments, which stand out at right angles from the surface of the leaf. Touch one of them with the end of a fine human hair, and the lobes of the leaf instantly close together in virtue of an act of contraction of part of their substance, just as the body of a snail contracts into its shell when one of its horns is irritated. The reflex action of the snail is the result of the presence of a nervous system in the animal. A molecular change takes place in the nerve of the tentacle, is propagated to the muscles by which the body is retracted, and causing them to contract, the act of retraction is brought about. Of course, the similarity of the acts does not necessarily involve the conclusion that the mechanism by which they are effected is the same, but it suggests a suspicion of their identity, which needs careful testing. The results of inquiries into the structure of the nervous system of animals converge toward the conclusion that the nerve fibers, which have been regarded as ultimate elements of nervous tissue, are not such, but are simply the visible aggregations of vastly more attenuated filaments, the diameter of which dwindles down to the limits of our present microscopic vision, greatly as these have been extended by modern improvements of the microscope, and that a nerve is, in its essence, nothing but a linear tract of specially modified protoplasm between 2 points of an organism, one of which is able to affect the other by means of the communication so established. Hence, it is conceivable that even the simplest living being may possess a nervous system. And the question whether plants are provided with a nervous system or not thus acquires a new aspect and presents the histologist and physiologist with a problem of extreme difficulty, which must be attacked from a new point of view and by the aid of methods which have yet to be invented. Thus, it must be admitted, he says again, that plants may be contractile and locomotive, that while locomotive, their movements may have as much appearance of spontaneity as those of the lowest animals, and that many exhibit actions comparable to those which are brought about by the agency of a nervous system in animals. And it must be allowed to be possible that further research may reveal the existence of something comparable to a nervous system in plants, so that I know not where we can hope to find any absolute distinction between animals and plants, unless we return to their mode of nutrition and inquire whether certain differences of a more occult character than those imagined to exist by Cuvier, and which certainly hold good for the vast majority of animals and plants, are of universal application. A bean may be supplied with water in which salts of ammonia and certain other mineral salts are dissolved in due proportion, with atmospheric air containing its ordinary minute dose of carbonic acid, and with nothing else but sunlight and heat. Under these circumstances, unnatural as they are, with proper management, the bean will thrust forth its radicle and its plumule. The former will grow down into roots, the latter grow up into the stem and leaves of a vigorous bean plant, and this plant will, in due time, flower and produce its crop of beans just as if it were grown in the garden or in the field. The weight of the nitrogenous protein compounds, of the oily, starchy, saccharine, and woody substances contained in the full-grown plant and its seeds will be vastly greater than the weight of the same substances contained in the bean from which it sprang. But nothing has been supplied to the bean save water, carbonic acid, ammonia, potash, lime, iron, and the like in combination with phosphoric, sulfuric, and other acids. Neither protein, nor fat, nor starch, nor sugar, nor any substance in the slightest degree resembling them has formed part of the food of the bean. But the weights of the carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur, and other elementary bodies contained in the bean plant and in the seeds which it produces are exactly equivalent to the weights of the same elements which have disappeared from the materials supplied to the bean during its growth. Whence it follows that the bean has taken in only the raw materials of its fabric and has manufactured them into bean stuffs. The bean has been able to perform this great chemical feat by the help of its green coloring matter, or chlorophyll, for it is only the green parts of the plant which, under the influence of sunlight, have the marvelous power of decomposing carbonic acid, setting free the oxygen, and laying hold of the carbon which it contains. In fact, the bean obtains two of the absolutely indispensable elements of its substance from two distinct sources. The watery solution in which its roots are plunged contains nitrogen but no carbon. The air to which the leaves are exposed contains carbon, but its nitrogen is in the state of a free gas, in which condition the bean can make no use of it, and the chlorophyll is the apparatus by which the carbon is extracted from the atmospheric carbonic acid, the leaves being the chief laboratories in which this operation is effected. End of section 10. Recording by Linda Johnson. Section 11 of The Science History of the Universe, Volume 5. This is a LibriVox recording. All LibriVox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. The Science History of the Universe, Volume 5, edited by Francis Rolt Wheeler. Biology, Chapter 7, Organic Functions, Part 2. The great majority of conspicuous plants are, as everybody knows, green, and this arises from the abundance of their chlorophyll. The few which contain no chlorophyll and are colorless are unable to extract the carbon which they require from atmospheric carbonic acid, and lead a parasitic existence upon other plants. But it by no means follows, often as the statement has been repeated, that the manufacturing power of plants depends on their chlorophyll and its interaction with the rays of the sun. On the contrary, it is easily demonstrated, as Pasteur first proved, that the lowest fungi, devoid of chlorophyll or of any substitute for it as they are, nevertheless possess the characteristic manufacturing power of plants in a very high degree. Only it is necessary that they should be supplied with a different kind of raw material. As they cannot extract carbon from carbonic acid, they must be furnished with something else that contains carbon. Tartaric acid is such a substance, and if a single spore of the commonest and most troublesome of molds, penicillium, be sown in a saucerful of water in which tartrate of ammonia with a small percentage of phosphates and sulfates is contained and kept warm, whether in the dark or exposed to light, it will in a short time give rise to a thick crust of mold, which contains many million times the weight of the original spore in protein compounds and cellulose. Thus, we have a very wide basis of fact for the generalization that plants are essentially characterized by their manufacturing capacity, by their power of working up mere mineral matters into complex organic compounds. Contrariwise, there is a no less wide foundation for the generalization that animals, as Cuvier put it, depend directly or indirectly upon plants for the material of their bodies. That is, either they are herbivorous or they eat other animals which are herbivorous. But for what constituents of their bodies are animals thus dependent upon plants? Certainly not for their horny matter, nor for chondrin, the proximate chemical element of cartilage, nor for gelatin, nor for syntonin, the constituent of muscle, nor for their nervous or biliary substances, nor for their amyloid matters, nor necessarily for their fats. It can be experimentally demonstrated that animals can make these for themselves, but that which they cannot make, but must in all known cases obtain directly or indirectly from plants, is the peculiar nitrogenous matter, protein. Thus, the plant is the ideal proletariat of the living world, the worker who produces, the animal, the ideal aristocrat, who mostly occupies himself in consuming, after the manner of that noble representative of the line of Zadarm, whose epitaph is written in Sartor Resartus. Here is our last hope of finding a sharp line of demarcation between plants and animals, for, as I have already hinted, there is a border territory between the two kingdoms, a sort of no-man's land, the inhabitants of which certainly cannot be discriminated and brought to their proper allegiance in any other way. Some months ago, Professor Tyndall asked me to examine a drop of infusion of hay placed under an excellent and powerful microscope, and to tell him what I thought some organisms visible in it were. I looked and observed, in the first place, multitudes of bacteria moving about with their ordinary intermittent spasmodic wriggles. As to the vegetable nature of these, there is now no doubt. Not only does the close resemblance of the bacteria to unquestionable plants, such as the oscillatoria, and the lower forms of fungi justify this conclusion, but the manufacturing test settles the question at once. It is only needful to add a minute drop of fluid containing bacteria to water in which tartrate, phosphate, and sulfate of ammonia are dissolved, and in a very short space of time the clear fluid becomes milky by reason of their prodigious multiplication, which of course implies the manufacture of living bacterium stuff out of these merely saline matters. But other active organisms, very much larger than the bacteria, attaining in fact the comparatively gigantic dimensions of 1/3000th of an inch or more, incessantly crossed the field of view. Each of these had a body shaped like a pear, the small end being slightly incurved and produced into a long curved filament, or cilium, of extreme tenuity. Behind this, from the concave side of the incurvation, proceeded another long cilium, so delicate as to be discernible only by the use of the highest powers and careful management of the light. In the center of the pear-shaped body, a clear, round space could occasionally be discerned, but not always, and careful watching showed that this clear vacuity appeared gradually, and then shut up and disappeared suddenly at regular intervals. Such a structure is of common occurrence among the lowest plants and animals, and is known as a contractile vacuole. The little creature thus described sometimes propelled itself with great activity, with a curious rolling motion, by the lashing of the front cilium, while the second cilium trailed behind. Sometimes it anchored itself by the hinder cilium, and was spun around by the working of the other, its motions resembling those of an anchor buoy in a heavy sea. Sometimes, when two were in full career toward one another, each would appear dexterously to get out of the other's way. Sometimes a crowd would assemble and jostle one another with as much semblance of individual effort as a spectator on the Grand Moulet might observe with a telescope among the specks representing men in the valley of Chamonix. The spectacle, though always surprising, was not new to me, so my reply to the question put to me was that these organisms were what biologists call monads, and though they might be animals, it was also possible that they might, like the bacteria, be plants. My friend received my verdict with an expression which showed a sad want of respect for authority. He would as soon believe that a sheep was a plant. Naturally piqued by this want of faith, I have thought a good deal over the matter, and as I still rest in the lame conclusion I originally expressed, must even now confess that I cannot certainly say whether this creature is an animal or a plant. Thus, in the question of making proteid, there are all gradations from plants able to make proteid from inorganic matter to plants which are as much animal as vegetable in structure, but are animal in the dependence on other organisms for their food. The singular circumstance observed by Meyer that the torula of yeast, though an indubitable plant, still flourishes most vigorously when supplied with the complex nitrogenous substance pepsin, the probability that the potato blight is nourished directly by the protoplasm of the potato plant, and the wonderful facts which have recently been brought to light respecting insectivorous plants all favor this view and tend to the conclusion that the difference between animal and plant is one of degree rather than of kind, and that the problem whether in a given case an organism is an animal or a plant may be essentially insoluble. This conception that animals and plants differ not in kind has been of great importance in the development of biology as a unified science of life. But although the differences in the organic functions are of degree rather than of kind, it is true that the differences of degree are clear enough, except in certain unicellular organisms, which are on the borderline, to enable us to distinguish between animals and plants and to make it convenient to divide physiology, the science of function, into plant and animal physiology. These differences will be clearer by adding to Huxley's general comparison of plants and animals a concrete comparison of an animal and a plant. For this purpose, one of Huxley's most famous students, T. Jeffrey Parker, chose the unicellular animal amoeba and the unicellular plant Haematococcus, also known as Sphaerella. Both of these live in water. In their nutrition, there are some differences, for amoeba can take in solid food, other small plants and animals, formed of protoplasm, as complex as its own. These it breaks up by means of digestive juices into soluble proteids and then recombines them to form its own protoplasm. Haematococcus has not this ability to take in solid food. It never feeds in the ordinary sense of the word. Nevertheless, it must take in food in some way or other, or the decomposition of its protoplasm would soon bring it to an end. The water in which it lives is never pure, but always contains certain mineral salts in solution, especially nitrates, ammonia salts, and often sodium chloride or common table salt. These salts, being crystalloids, can and do diffuse into the water of the organism, so that we may consider its protoplasm to be constantly permeated by a very weak saline solution, the most important elements contained in which are oxygen, hydrogen, nitrogen, potassium, sodium, calcium, sulfur, and phosphorus. It must be remarked, however, that the diffusion of these salts does not take place in the same uniform manner as it would through parchment or other dead membrane. The living protoplasm has the power of determining the extent to which each constituent of the solution shall be absorbed. If water containing a large quantity of Haematococcus is exposed to sunlight, minute bubbles are found to appear in it, and these bubbles, if collected and properly tested, are found to consist largely of oxygen. Accurate chemical analysis has shown that this oxygen is produced by the decomposition of the carbon dioxide contained in solution in rainwater, and indeed in all water exposed to the air, the gas, which is always present in small quantities in the atmosphere, being very soluble in water. As the carbon dioxide is decomposed in this way, its oxygen being given off, it is evident that its carbon must be retained. As a matter of fact, it is retained by the organism, but not in the form of carbon. In all probability, a double decomposition takes place between the carbon dioxide absorbed and the water of organization, the result being the liberation of oxygen in the form of gas and the simultaneous production of some extremely simple form of carbohydrate, i.e. some compound of carbon, hydrogen, and oxygen with a comparatively small number of atoms to the molecule. The next step seems to be that the carbohydrate thus formed unites with the ammonia salts or the nitrates absorbed from the surrounding water, the result being the formation of some comparatively simple nitrogenous compound. Then further combinations take place, substances of greater and greater complexity are produced, sulfur from the absorbed sulfates enters into combination, and proteids are formed. From these, finally, fresh living protoplasm arises. From the foregoing account, which only aims at giving the very briefest outline of a subject as yet imperfectly understood, it will be seen that, as in amoeba, the final result of the nutritive process is the manufacture of protoplasm, and that this result is attained by the formation of various substances of increasing complexity. But it must be noted that the steps in the process of constructive metabolism are widely different in the two cases. In amoeba, we start with living protoplasm, that of the prey, which is killed and broken up into diffusible proteids, these being afterward recombined to form new molecules of the living protoplasm of amoeba, so that the food of amoeba is, to begin with, as complex as itself, and is broken down by digestion into simpler compounds, these being afterward recombined into more complex ones. In Haematococcus, on the other hand, we start with extremely simple compounds such as carbon dioxide, water, nitrates, sulfates, etc. Nothing which can be properly called digestion, i.e. a breaking up and dissolving of the food, takes place, but its various constituents are combined into substances of gradually increasing complexity, protoplasm as before being the final result. To express the matter in another way, amoeba can only make protoplasm out of proteids already formed by some other organism. Haematococcus can form it out of simple liquid and gaseous inorganic materials. Speaking generally, it may be said that these two methods of nutrition are respectively characteristic of the two great groups of living things. Animals require solid food containing ready-made proteids and cannot build up their protoplasm out of simpler compounds. Green plants, i.e. all the ordinary trees, shrubs, weeds, etc., take only liquid and gaseous food and build up their protoplasm out of carbon dioxide, water, and mineral salts. The first of these methods of nutrition is conveniently distinguished as holozoic, or wholly animal, the second as holophytic, or wholly vegetal. It is important to note that only those plants or parts of plants in which chlorophyll is present are capable of holophytic nutrition. Whatever may be the precise way in which the process is effected, it is certain that the decomposition of carbon dioxide, which characterizes this form of nutrition, is a function of chlorophyll, or to speak more accurately, of chromatophores, since there is reason for thinking that it is the protoplasm of these bodies and not the actual green pigment which is the active agent in the process. Moreover, it must not be forgotten that the decomposition of carbon dioxide is carried on only during daylight, so that organisms in which holophytic nutrition obtains are dependent upon the sun for their very existence. While amoeba derives its energy from the breaking down of the proteids in its food, the food of Haematococcus is too simple to serve as a source of energy, and it is only by the help of sunlight that the work of constructive metabolism can be carried on. This may be expressed by saying that Haematococcus, in common with other organisms containing chlorophyll, is supplied with kinetic energy in the form of light or radiant energy directly by the sun. As in amoeba, destructive metabolism is constantly going on, side by side with constructive. The protoplasm becomes oxidized, water, carbon dioxide, and nitrogenous waste matters being formed and finally got rid of. Obviously, then absorption of oxygen must take place, or in other words, respiration must be one of the functions of the protoplasm of Haematococcus, as of that of amoeba. In many green, i.e. chlorophyll-containing plants, this has been proved to be the case. Respiration, i.e. the taking in of oxygen and giving out of carbon dioxide, is constantly going on, but during daylight is obscured by the converse process, the taking in of carbon dioxide for nutritive purposes and the giving out of the oxygen liberated by its decomposition. In darkness, when this latter process is in abeyance, the occurrence of respiration is more readily ascertained. End of section 11. Recording by Linda Johnson. Section 12 of The Science History of the Universe, Volume 5. This is a LibriVox recording. All LibriVox recordings are in the public domain. For more information or to volunteer, please visit librivox.org. The Science History of the Universe, Volume 5, edited by Francis Rolt Wheeler. Biology, Chapter 8. Life Processes, Part 1. Nutrition thus, as has been pointed out, makes it possible to classify most organisms as animals or plants. Yet there are many unicellular forms in which both kinds of nutrition go on at the same time. That is, the forms may possess a mouth for the ingestion of solid food and green coloring matter, chlorophyll, for the manufacture of starchy food from gaseous matter. Many of the lowest forms of life have long been puzzles, and the beginner in biological study is surprised to find them described in textbooks of both botany and zoology. The fact is that they are on the borderline, are neither plants nor animals, but simply organisms. Since they cannot be classified, it is necessary that they be listed both under botany and zoology in order to make sure that they will not be omitted entirely. Because of these uncertain forms of life, Haeckel proposed once to include all one-celled animals and plants in a third kingdom to be called Protista, meaning the first of all life. Parker's definition of animals and plants, based on the foregoing considerations, is convenient for distinguishing between animals and plants in all cases except the doubtful unicellular forms. He says, animals are organisms of fixed and definite form, in which the cell body is not covered with a cellulose wall. They ingest solid proteinaceous food, their nutritive processes result in oxidation, they have a definite organ of excretion, and are capable of automatic movement. Plants are organisms of constantly varying form, in which the cell body is surrounded by a cellulose wall. They cannot ingest solid food, but are nourished by a watery solution of nutrient materials. If chlorophyll is present, the carbon dioxide of the air serves as a source of carbon. Nitrogen is obtained from simple salts, and the nutritive processes result in deoxidation. If chlorophyll is absent, carbon is obtained from sugar or some similar compound, nitrogen either from simple salts or from proteids, and the process of nutrition is one of oxidation. There is no special excretory organ, and, except in the case of certain reproductive bodies, there is usually no locomotion. The important point to recognize is that these boundaries are artificial, and that there are no scientific frontiers in nature. As in the liquefaction of gases, there is a critical point at which the substance under experiment is neither gaseous nor liquid. As in a mountainous country, it is impossible to say where mountain ends and valley begins. As in the development of an animal, it is futile to argue about the exact period when, for instance, the egg becomes a tadpole or the tadpole a frog, so in the case under discussion. The distinction between the higher plants and animals is perfectly sharp and obvious, but when the two groups are traced downward, they are found gradually to merge, as it were, into an assemblage of organisms which partake of the characters of both kingdoms, and cannot, without a certain violence, be either included in or excluded from either. When any given protist has to be classified, the case must be decided on its individual merits. The organism must be compared in detail with all those which resemble it closely in structure, physiology, and life history, and then a balance must be struck, and the doubtful form placed in the kingdom with which it has, on the whole, most points in common. It will no doubt occur to the reader that, on the theory of evolution, the fact of the animal and vegetable kingdoms being related to one another like two trees united at the roots may be accounted for by the hypothesis that the earliest organisms were protists, and that from them animals and plants were evolved along divergent lines of descent. And in this connection, the fact that some bacteria, the simplest organisms known and devoid of chlorophyll, may flourish in solutions wholly devoid of organic matter is very significant. The lower plants and animals referred to above are so far from everyday observation, and hence so unfamiliar, that to most people the comparison made will mean little in terms of ordinary green flowering plants and common vertebrate animals. In order to emphasize the fundamental similarity of organic function in higher and lower animals and plants, let us compare any higher plant, e.g. a bean plant, with a higher animal, e.g. frog, or even man. In each, the life is the sum total of a series of definite processes: nutrition or food supply, circulation, metabolism, excretion, oxygenation, part of respiration, movement, irritability, nervous activity, and reproduction. In turn, these will be compared for the animal and the plant, following in part the comparisons of certain animals and plants by Sedgwick and Wilson and others. These comparisons will, however, be translated into terms applicable to any species of higher plants or animals. In the nutrition of the animal, the most essential and characteristic part of the food supply is derived from vegetable or animal matter in the form of various organic compounds, of which the most important are proteids, protoplasm, albumin, etc., carbohydrates, starch, cellulose, and fats. These materials are used by the animal in the manufacture of new protoplasm to take the place of that which has been used up. It is, however, impossible for the animal to build these materials directly into the substance of its own body. They must first undergo certain preparatory chemical changes known collectively as digestion, and only after the completion of this process can all the food be absorbed into the circulation. For this purpose, the food is taken not into the body proper, but into a kind of tubular chemical laboratory called the alimentary canal, through which it slowly passes, being subjected meanwhile to the action of certain chemical substances or reagents known as digestive ferments. These substances, which are dissolved in a watery liquid to form the digestive fluid, are secreted by the walls of the alimentary tube. Through their action, the solid portions are liquefied and the food is rendered capable of absorption into the body proper. The food supply of the higher plant, like that of the animal, is the source of the required matter and energy, but, unlike that of the animal, it is not chiefly an income of foods, but only of the raw materials of food. Matter enters the plant in the liquid or gaseous form by diffusion, both from the soil through the roots, liquids, and from the atmosphere through the leaves, gases. We have here the direct absorption into the body proper of foodstuffs precisely as the animal takes in water and oxygen. Energy enters the plant, to a small extent, as the potential energy of foodstuffs, but comes in principally as the kinetic energy of sunlight absorbed in the leaves. Of the substances, the solids, salts, etc., must be dissolved in water before they can be taken in. Water and dissolved salts continually pass by diffusion from the soil into the roots, where, together, they constitute the sap. The sap travels throughout the whole plant, the main, though not the only cause of movement, being the constant transpiration, evaporation, of watery vapor from the leaves, especially through the stomata. The gaseous matters, carbon dioxide, oxygen, nitrogen, enter the plant mainly by diffusion from the atmosphere, are dissolved by the sap in the leaves and elsewhere, and thus may pass to every portion of the plant. The green plant owes its power of absorbing the energy of sunlight to the chlorophyll bodies or chromatophores, for plants which, like fungi, etc., are devoid of chlorophyll, are unable thus to acquire energy. Entering the chlorophyll bodies, the kinetic energy of sunlight is applied to the decomposition of carbon dioxide and water. After passing through manifold but imperfectly known processes, the elements of these substances finally reappear as starch, often in the form of granules embedded in the chlorophyll bodies and free oxygen, most of which is returned to the atmosphere. Thus, the leaf of a green plant in the light is continually absorbing carbon dioxide and giving forth free oxygen. Carbon dioxide and water contain no potential energy, since the affinities of their constituent elements are completely satisfied. Starch, however, contains potential energy, since the molecule is relatively unstable, i.e., capable of decomposition into simpler, stabler molecules in which stronger affinities are satisfied. And this is due to the fact that in the manufacture of starch in the chlorophyll bodies, the kinetic energy of sunlight was expended in lifting the atoms into position of vantage, thus endowing them with energy of position. In this way, some of the radiant and kinetic energy of the sun comes to be stored up as potential energy in the starch. In short, the green plant is able, by cooperation with sunlight, to use simple raw materials, carbon dioxide, water, oxygen, etc., poor in energy or devoid of it, and out of them to manufacture food, i.e., complex compounds rich in available potential energy. This power is possessed by green plants alone, all other organisms being dependent for energy upon the potential energy of ready-made food. This must, in the first instance, be provided for them by green plants, and hence, without chlorophyll-bearing plants, animals and colorless plants as well apparently could not long exist. The plant absorbs also a small amount of kinetic energy independently of the sunlight in the form of heat. This, however, is probably not a source of vital energy, but only contributes to the maintenance of the body temperature. Food, starch, thus produced in the green leaves of higher plants and the inorganic foods, water, nitrates or nitrates and various mineral substances in solution in water, furnish the materials and energy required for the life and growth of the plant. The circulatory system distributes these foods. In animals, foods prepared for absorption in the stomach and intestine by digestion are absorbed by the circulating liquids, blood and lymph, and transported to all cells of the animal body. In the plant, the inorganic matter in water from the soil are absorbed by the roots and carried up definite tubes in the woody part of the stem. The causes of this ascent are not clear, but root pressure due to osmosis, capillary action, and evaporation from the leaves are factors. Just as the solid food of animals must be digested in preparation for absorption, so starch manufactured in the leaves must be digested, dissolved, before it can be transported. This is done by diastase, an enzyme of plant cells. The change is from starch to a sugar capable of diffusion. Dissolved in water, the sugar is transported down delicate tubes, chiefly in the growing bark region of the stem. It is clear that there are upward and downward currents of water containing food, comparable to blood of an animal, but no system of complete circulation as in the blood vessels of a higher animal. However, the result in distributed food is the same in the plant and in the animal. In the cells, the foods undergo metabolic changes. In an animal, the foods in the circulating liquids, blood and lymph, are selected and absorbed by the cells. Only proteid foods form new protoplasm, and even of proteids, only a limited amount, 75 to 100 grams a day for a man, is built into new protoplasm. The excess undergoes oxidation and forms nitrogen excretions. The foods containing only the elements carbon, hydrogen, and oxygen, fats and carbohydrates, are directly oxidized to excretions and, lacking nitrogen, cannot serve for making new animal protoplasm. Fat and carbohydrate foods, then, never become living matter. They may be stored, especially as fat, until needed for oxidation to supply energy. The building up of the protoplasm from proteids is anabolism, constructive metabolism. The destruction of protoplasm, excess proteids, or the fat and carbohydrate foods, is catabolism, destructive metabolism. Catabolism is probably due to enzyme action, but the final result is chiefly carbon dioxide and water, which could be derived by the ordinary chemical evolution of protoplasm, proteid, sugar, starch, or fats. In the plant, starch, as has been seen, is first formed in the chlorophyll bodies, but the formation of starch, all important as it is after all only the manufacture of food as a preliminary to the real processes of nutrition. These processes must take place everywhere in ordinary protoplasm, for it is here that oxidation occurs, and the need for a renewal of matter and energy consequently arises. Sooner or later, the starch grains are changed into a kind of sugar, glucose, which, unlike starch, dissolves in the sap and may thus be easily transported to all parts of the plant. Wherever there is need for new protoplasm, whether to repair previous waste or to supply materials for growth, after absorption into the cells, the elements of the starch or glucose are, by the living protoplasm, in some unknown way, combined with nitrogen and sulfur, probably also with salts, water, etc., to form proteid matter. The particles of this newly formed compound are incorporated into the protoplasm. If a larger quantity of starch is formed in the chlorophyll bodies than is immediately needed by the protoplasm for purposes of repair or growth, it may be reconverted into starch after journeying as glucose through the plant and be laid down as reserve starch in the cells of root or stem or elsewhere. Apparently, when this reserve supply is finally needed at any point in the plant, it is again changed to glucose and transported thither. It is probable that new leaves and new tissues generally are always formed in part from this reserve starch. In the plant, as in the animal, metabolism must consist of anabolic and catabolic processes. The construction in the cells of new proteid from the absorbed carbohydrate and the materials from the soil is true anabolism. It is also clear that catabolism, or oxidation for the liberation of energy, occurs as in animals, but this process is slower. Probably, foods containing carbon, hydrogen, and oxygen are the sources of energy in the higher plants as in animals. In both plants and animals, simple waste substances result from the catabolic processes in the cells. In the animal, carbon dioxide, water, and nitrogen compounds are the chief excretions. They are absorbed by the circulating liquids and carried to the eliminating organs, lungs and kidneys chiefly, for elimination. In the higher plants, the excretions are carbon dioxide, which escapes through the epidermis of root, stem, and leaf, and through the stomata, water which is lost by evaporation, especially from the leaf surface through the stomata, excretions which are lost by osmosis through the roots, and the accumulated but useless mineral substances which are eliminated by leaf fall. In both animals and plants, oxygen is essential to the catabolic part of metabolism. Hence, oxygen must be supplied to the cells. Oxygenation is the term used to denote the oxygen-supplying part of respiration. The other part of respiration, elimination of carbon dioxide, has been treated under excretions. In the animal, oxygen is absorbed by the blood, in excess by the hemoglobin of the red cells of the blood, and later is absorbed from the blood and lymph by all the living cells. In the plant also, oxygen is absorbed through the epidermis and stomata from the air. This process is, however, obscured during the day because of the oxygen freed in the manufacture of starch which goes on at that time. Probably this freed oxygen is used for the purpose of oxygenation, but more is freed in the photosynthetic process than is needed for oxygenation, and hence the excess oxygen is eliminated, while starch manufacture is in process. In comparing a higher animal and a green plant, confusion must be avoided regarding the part played by oxygen and carbon dioxide in true respiration with the part played by the same substances in starch formation, photosynthesis. In non-green plants like the Indian pipe and mushrooms, the breathing of oxygen and the excretion of carbon dioxide are as in the animal. This is true also of green plants in darkness and even in the light of all parts of green plants except the chlorophyll bodies. These constitute a sort of extra mechanism enabling green plants to make their own carbohydrate food. Imagine a higher animal with an attachment for turning the carbon dioxide and water excreted back to starch usable as food, and the comparison of the green plant and the animal would be complete. End of section 12. Recording by Linda Johnson. Section 13 of The Science, History of the Universe, Volume 5. This is a LibriVox recording. All LibriVox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. The Science, History of the Universe, Volume 5, edited by Francis Rolt Wheeler. Biology, Chapter 8, Life Processes, Part 2. The power of movement, or locomotion, is curiously thought of as peculiar to animals, but biologists know dozens of examples of movement in plants. Some of the lower plants possess the power of locomotion, and even in plants as high as mosses and ferns, there is a locomotion of the male germ cells. Among the flowering plants, there is no actual locomotion, but there are numerous forms of movement. Most striking, perhaps, are the so-called sleep movements of plants, by means of which the leaves of some plants, e.g. the oxalis, bean, clover, locust, and others, can assume decidedly different positions at night from those which they occupy by day. The movements of the sensitive plant, mimosa, when shocked by touch or in other ways, and the movements of the leaves of the Venus flytrap, Dionia, which close with a quick jerk when the sensitive hairs with which they are bordered are stimulated. It is in nervous functions that the most striking difference between the animal and the plant is to be found. Plants have no true nervous system. Nevertheless, irritability or response to stimulus is well marked in many plants. It would carry us far beyond the limits of this volume to trace satisfactorily the powers of plants to respond to stimuli of gravity, light, heat, water, electricity, chemicals, contact, injury, and so on. Suffice it to say that the total effect of such responses is for the plant equal to that of the nervous system for the animal. Reproduction is a most important function. The life of every organic species runs in regularly recurring cycles, for every individual life has its limit. In youth, the constructive processes preponderate over the destructive, and the organism grows. The normal adult attains a state of apparent physiological balance in which the processes of waste and repair are approximately equal. Sooner or later, however, this balance is disturbed. Even though the organism escapes every injury or special disease, the constructive process falls behind the destructive. Old age ensues, and the individual dies from sheer inability to live. Why the vital machine should thus wear out is a mystery, but that it has a definite cause and meaning is indicated by the familiar fact that the span of natural life varies with the species. Man lives longer than the dog, the elephant longer than man. It is a wonderful fact that living things have the power to detach from themselves portions or fragments of their own bodies endowed with fresh powers of growth and development and capable of running through the same cycle as the parent. There is, therefore, an unbroken material, protoplasmic, continuity from one generation to another that forms the physical basis of inheritance and upon which the integrity of the species depends. As far as known, living things never arise save through this process. In other words, every mass of existing protoplasm is the last link in an unbroken chain that extends backward in the past to the first origin of life. The detached portions of the parent that are to give rise to offspring are sometimes masses of cells, as in the separation of branches or buds among plants, but more commonly they are single cells known as germ cells, like the eggs of animals and the spores of ferns and mosses. Only the germ cells, which may conveniently be distinguished from those forming the rest of the body or the somatic cells, escape death, and that only under certain conditions. All forms of reproduction fall under one or the other of two heads, viz. Agamogenesis, asexual reproduction, or gamogenesis, sexual reproduction. In the former case, the detached portion, which may be either a single cell or a group of cells, has the power to develop into a new individual without the influence of other living matter. In the latter, the detached portion, in this case always a single cell, ovum, oosphere, etc., is acted upon by a second portion of living matter, likewise a single cell, which in most cases has been detached from the body of another individual. The germ is called the female germ cell, the cell acting upon it the male germ cell, and in the sexual process, the two fuse together, fertilization, impregnation, to form a single new cell endowed with the power of developing into a new individual. In some organisms, e.g. the yeast plant and bacteria, only agamogenesis has been observed. In others, e.g. vertebrates, only gamogenesis. In others still, both processes take place, as in many higher plants. The earthworm, for example, is not known to multiply by a natural process of agamogenesis. It possesses in a high degree, however, the closely related power of regeneration. For if a worm be cut transversely into two pieces, the anterior piece will usually make good or regenerate the missing portion, while the posterior piece may regenerate the anterior region. Thus, the worm can, to a certain limited extent, be artificially propagated like a plant by cuttings, a process closely related to true agamogenesis. Its usual and normal mode of reproduction is by gamogenesis, that is, by the formation of male germ cells, spermatozoa, and female germ cells, ova. In higher animals, the two kinds of germ cells are produced by different individuals of opposite sex. The earthworm, on the contrary, is hermaphrodite or bisexual. Every individual is both male and female, producing both eggs and spermatozoa. As in the animal, so in the plant. Whether the individual dies or not, ample provision against the death of the race is made in the act of reproduction. Although reproduction appears to be useless to the individual, and even entails upon it serious annual losses of matter and energy, yet to this function every part of the plant directly or indirectly contributes. The reproductive germs are carefully prepared, are provided with a stock of food sufficient for the earliest stages of development, and are endowed with the peculiar powers and limitations of each species which influence their life history at every step and are, by them, transmitted in turn to their descendants. They are living portions of the parent, detached for reproductive purposes, and they contain a share of protoplasm directly descended from the original protoplasm from which the parent came. In short, reproduction is the supreme function of the plant. As in the animal, reproduction in the higher plants is by the process of gamogenesis. Male germ cells and female germ cells are formed in separate organs. In many of the higher plants, these organs are both found in the same individual, but often they are produced by different individuals of opposite sex. Agamogenesis, or asexual reproduction, also takes place in many higher plants by means of runners, buds, bulbs, tubers, etc. But in most of these cases, provision is made for sexual reproduction also. In relation to the environment, animals and plants are masses of living matter occupying definite positions in space and time and existing amid certain definite and characteristic physical surroundings which constitute their environment. As ordinarily understood, the term environment applies only to the immediate surroundings of the animal and plant. Strictly speaking, however, the environment includes everything that may in any manner act upon the organisms, that is, the whole universe outside them. For they are directly and profoundly affected by rays of light and heat that travel to them from the sun. They are extremely sensitive to the alternations of day and night and the seasons of the year. They are acted on by gravity, and to all these, as well as to more immediate influence, they make definite responses. The body of the animal is a complicated piece of mechanism constructed to perform certain definite actions. But every one of these actions is, in one way or another, dependent upon the environment and directly or indirectly relates to it. At every moment of its existence, the organism is acted on by its environment. At every moment it reacts upon the environment, maintaining with it a constantly shifting state of equilibrium which finally gives way only when the life of the animal draws to a close. The action of the environment upon the animal has been sufficiently stated. It remains to point out the changes worked by the animal on the environment. These changes are of two kinds: mechanical or physical, and chemical. The general effect of the metabolism of the animal is the destruction by oxidation of organic matter. That is, matter originally taken from the environment in the form of complex proteids, fats, and carbohydrates is returned to it in the form of simpler and more highly oxidized substances, of which the most important are carbon dioxide and water, both inorganic substances. This action furthermore is accompanied by a dissipation of energy, that is, a conversion of potential into kinetic energy. On the whole, therefore, the action of the animal upon the environment is that of an oxidizing agent, a reducer of complex compounds to simpler ones, and a dissipator of energy. The actions of the environment upon the plant have been sufficiently dwelt upon. It still remains, however, to consider the actions of the plant upon the environment. These are partly physical, but mainly chemical. By pushing its stems and leaves into the air and slowly thrusting its roots through the soil, the atmosphere and the earth are alike displaced. But it is by its chemical activity that it most profoundly affects its environment. Absorbing from the latter water, salts, carbon dioxide, and other simple substances, as well as sunlight, it produces with them a remarkable metamorphosis. It manufactures from them, as raw materials, organic matter in the shape of starch, fats, and even proteids. These it gives back to the environment in some measure during life, and surrenders wholly after sudden death. But the most striking fact is that the plant is on the whole constructive and capable of producing and accumulating compounds rich in energy, and in this respect it is unlike the animal. Thus, while animals are destroyers of energized compounds, green plants are producers of them. Animals, therefore, in the long run, are absolutely dependent on plants, and animals and colorless plants alike upon green plants. But it must never be forgotten that most plants are enabled to manufacture organic from inorganic matter by virtue of the chlorophyll which they contain. Without this, they are powerless in this respect. It is evident that to the superficial observer, the plant and animal seem to have little or nothing in common, except that both are what we call alive. But whoever has studied the preceding pages must have perceived beneath manifold differences of detail a fundamental likeness between the plant and animal, not only in the substantial identity of the living matter in the two, but also in the construction of their bodies and in the processes by which they come into existence. Each arises from a single cell, to quote Sedgwick and Wilson, which is the result of the union of two differently constituted cells, male and female. In both, the primary cell multiplies and forms a mass of cells, at first nearly similar, but afterward differentiated in various directions to enable them to perform different functions, i.e., to effect a physiological division of labor. In both, the tissues thus provided are associated more or less closely into distinct organs and systems, among which the various operations of the body are distributed. And in both, the ultimate goal of individual existence is the production of germ cells, which form the starting point of new and similar cycles. This fundamental likeness extends also to most of the actions, physiology, of the two organisms. Both possess the power of adapting themselves to the environments in which they live. Both take in various forms of matter and energy from the environment, build them up into their own living substance, and finally break down this substance more or less completely into simpler compounds by processes of internal combustion, setting free by this action the energy which maintains their vital activity. And, sooner or later, both give back to the environment the matter and energy which they have taken from it. In other words, both effect an exchange of matter and of energy with the environment. Nevertheless, the plant and the animal differ. They differ widely in form, and the plant is fixed and relatively rigid, while the animal is flexible and mobile. The body of the plant is relatively solid. That of the animal contains numerous cavities. The plant absorbs matter directly through the external surface. The animal partly through the external and partly through an internal, elementary surface. The plant is able to absorb simple chemical compounds from the air and earth and kinetic energy from sunlight. The animal absorbs, for the most part, complex chemical compounds and makes no nutritive use of the sun's kinetic energy. By the aid of this energy, the plant manufactures starch from simple compounds, carbon dioxide, and water. The animal lacks this power. The plant can build up proteids from the nitrogenous and other compounds of its food. And by manufacturing proteids within its living substance, the plant is relieved of the necessity of carrying on a process of digestion in order to render them diffusible for entrance into the body. Still, great as these differences appear to be at first sight, all of them, with a single exception, fade away upon closer examination. This exception is the power of making foods. Plants and animals differ in form because their mode of life differs, but a wider study of biology reveals the existence of innumerable animals, corals, sponges, hydroids, etc., which have a close superficial resemblance to plants, and of many plants which resemble animals, not only in form, but also in possessing the power of active locomotion. The stomach of the animal, as shown by its development, is really a part of the general outer surface, which is folded into the body, and the animal, like the plant, therefore, really absorbs its income over its whole surface. Oxygen through the general outer surface, other food matters through the infolded alimentary surface. In like manner, it is easy to show that not one of the differences between the plant and animal is fundamentally important, save the power of making foods. The animal must have complex, ready-made food, including proteid matter. So must the plant, but the plant is able to manufacture this complex food out of very simple compounds. In terms of energy, the animal requires ready-made food rich in potential energy. The plant, aided by the sun's energy, can manufacture food from matters devoid of energy. Hence, it appears, broadly speaking, that the plant, by the aid of solar energy, is constructive and stores up energy. The animal is destructive and dissipates energy. And this difference becomes of immense importance in view of the fact that it is true in this respect of all green plants as of all animals. Even this difference, great as it is partly bridged over by colorless plants like yeast, molds, bacteria, etc., which have no chlorophyll, are therefore unable to use the energy of light, and hence must have energized food. But these organisms do not, like animals, require proteid food, being able to extract all needful energy from the simpler fats, carbohydrates, and even from certain salts. When it is considered that the distinctive peculiarities of animals can thus be reduced to the sole characteristic of dependence on proteid food, it cannot be doubted that the difference between plants and animals is of immeasurably less importance than their fundamental likeness, the more so when it is kept in mind that each of the principles of organic function will be found to apply to all animals, including man himself, and to all plants, however complex they may be. End of section 13. Recording by Linda Johnson.
Section 14 of The Science, History of the Universe, Volume 5. This is a LibriVox recording. All LibriVox recordings are in the public domain. For more information or to volunteer, please visit librivox.org. Recording by Warren Cotty, Gurnee, Illinois. The Science, History of the Universe, Volume 5, edited by Francis Wolt Wheeler, Biology. Chapter 9, Origin of Species of Plants and Animals. There are, in the museums of the world at the present time, representatives of several 100,000, probably more than 1 million, kinds or species of plants and animals, and thousands of new species are being discovered and named each year. A single group of insects has classified under it more species than there are stars to be seen in the heavens with the unaided eye on a clear night. Aristotle knew about 500 kinds of animals, but a single new botanical or zoological work may now describe more than that number of species new to the records of science. Linnaeus in 1758 published the 10th edition of his Systema Naturae and named about 4,000 animals, and every year since 1864, the zoological record has listed 3 or 4 times this number of species, previously undescribed. Yet now, as in Linnaeus's time, it is certain that not half of the number of species of animal organisms is yet known. The 600,000, more or less, on the registers of science today are certainly far less than half of the millions which actually exist. In botany, the same conditions are to be found. There are fewer known species of plants than animals by half, and yet they are more easily preserved and handled, while the work of collection and investigation proceeds on a scale even more extensive. Yet it would be a bold statement to say that today half the species of plants that exist are known. All this refers to the forms now living, without reference to the host which composes their long ancestry, extending backward toward the dawn of creation. The species have come down through the geological ages, changing in form and function to meet the varying needs of changing environment. This enumeration takes no account of the still vaster myriads of forms, almost endlessly varied, which have perished utterly in the pressure of environment, leaving no trace in the line of descent. It is evident that variety in life is a factor in the history of the globe, that it may be expressed in terms of number of species, but that the actual range of variation is far greater than the number of species, and that if causes are to be judged by range of effects, in the origin of species must be found the operation of worldwide forces. The cooperation of great influences, far-reaching in time and space, as broad as the surface of the globe, and as enduring as its life. The cause of this amazing variety in life and the manner of accounting for origin of species are questions necessarily asked when attention is called to the existence of such vast numbers of species of plants and animals. In earlier chapters, the theories of the origin of life have been described, and proof that present living matter has descended from pre-existing living matter has been outlined. It is, in fact, accepted in modern science that there has been a continuity of descent from the first living matter to that of the present time. But the form or species in which life first originated is another subject, and almost as great. Such a subject must deal with the questions as to whether life first appeared in each species of animal or plant separately, or whether it began in simple protoplasm from which have been evolved the almost numberless species known today. It is quite clear that there are only 2 hypotheses in the field whereby it is possible so much as to suggest an explanation of the origin of species. Either all the species of plants and animals must have been supernaturally created, or else they must have been naturally evolved. There is no third hypothesis possible, for no one can rationally suggest that species have been eternal. It should be noticed that whichever of the 2 rival theories is entertained, the concern is not with any question touching the origin of life, but only with the origin of particular forms of life, that is to say, with the origin of species. The theory of descent starts from life as a datum already granted. How life itself came to be, the theory of descent, as such, is not concerned to show. Therefore, in the present discussion, the existence of life is taken as a fact, which does not fall within its range of debate. The history of biology in the 19th century will be famous because of the discussion of these two hypotheses, which attempt to account for the existence of the innumerable species of living things which inhabit the earth: the theory of creation and the theory of evolution. According to the theory of creation, all the individuals of every species existing at the present day, the tens of thousands of dogs, oak trees, amoeba, and whatnot, are derived by a natural process of descent from a single individual, or from a pair of individuals, in each case precisely resembling, in all essential respects, their existing descendants, which came into existence by a process outside the ordinary course of nature and known as creation. According to the rival theory, that of descent or organic evolution, every species existing at the present day is derived by a natural process of descent from some other species which lived in a former period of the world's history. If from generation to generation the individuals of any existing species could be traced back, on this hypothesis, their characters would be found gradually to change, until finally a period was reached at which the differences were so considerable as to necessitate the placing of the ancestral forms in a different species from their descendants at the present day. And in the same way, if the species of any one genus could be traced back, they would gradually approach one another in structure until they finally converged in a single species, differing from those now existing, but standing to all in a true parental relation. In regard to the present standing of these two theories, it should be stated that the theory of descent is now generally accepted by men of science and the theory of special creation rejected. In fact, no great naturalist since Agassiz has attacked the general theory, though some have debated many of its minor details. As David Starr Jordan has said, there is today no doubt in our minds of the truth, the actuality of descent. It is not the theory of descent. It is the fact, the law of descent, of which we talk and write. Organisms are blood-related. They are transformed, descended from one another. At this point, it will clarify some later considerations if it is emphasized that there is a great distinction to be drawn between the fact of evolution and the manner of it, or between the evidence of evolution as having taken place somehow and the evidence of the causes which have been concerned in the process. This most important distinction is frequently disregarded by popular writers on evolution, and therefore, in order to mark it as strongly as possible, it will be necessary to effect a complete separation between the evidence of evolution as a fact and the evidence as to its method. In other words, not until the evidence of organic evolution as a process, which somehow or another has taken place, has been fully considered, is it advisable to consider how it has taken place, or the causes which Darwin and others have suggested as having probably been concerned in this process. First, there is to be considered, therefore, the evidences pointing to the fact of organic descent, evolution of species of animals and plants, and later there will be outlined the trend of the enormous amount of investigation which has been and now, more actively than ever, is being done toward the solution of the problems concerned with the causes or the factors of evolution. In passing, it must be noted that while Darwin wrote both concerning the evidences of evolution and the manner or causes of evolution, it is not correct, as many authors assume, to regard the Darwinian theory or Darwinism as synonymous with the theory of descent or evolution. Rather, should the terms Darwinism and Darwinian theory be applied to the theory of natural selection, Darwin's great explanation of the cause of the origin of species by evolution or descent. The late Professor Cope of Philadelphia defines evolution in the broadest sense, including both organic and inorganic evolution, as follows. The doctrine of evolution may be defined as the teaching which holds that creation has been and is accomplished by the agency of the energies which are intrinsic in the evolving matter, and without the interference of agencies which are external to it. It holds this to be true of the combinations and forms of inorganic nature, and of those of organic nature as well, whether the intrinsic energies which accomplish evolution be forms of radiant or other energy only, acting inversely as the square of the distance, and without consciousness, or whether they be energies whose direction is affected by the presence of consciousness. The energy is a property of the physical basis of tridimensional matter, and is not outside of it. But a distinction must be made between organic and inorganic evolution. Professors Jordan and Kellogg have stated this most clearly in Evolution and Animal Life. Biological evolution and cosmic evolution are not the same. From the biological side, a certain objection must be made to this philosophical theory of universal or cosmic evolution. In organic and inorganic evolution, there is much in common, so far as conditions and results are concerned, but these likenesses belong to the realm of analogy, not of homology. They are not true identities because not arising from like causes. The evolution of the face of the earth forces parallel changes in organic life, but the causes of change in the two cases are in no respect the same. The forces or processes by which mountains are built or continents established have no homology with the forces or processes which transformed the progeny of reptiles into mammals or birds. Tendencies in organic development are not mystic purposes, but actual functions of actual organs. Tendencies in inorganic nature are due to the interrelations of mass and force. Whatever may be the final meanings attached to these terms or to the terms matter and energy, it is not clear that science has been really advanced through the conception of the essential unity of organic evolution and cosmic evolution. The relatively little the two groups of processes have in common has been overemphasized as compared with their fundamental differences. The laws which govern living matter are in a large extent peculiar to the process of living. Processes which are functions of organs cannot exist where there are no organs. The traits of protoplasm are shown only in the presence of protoplasm. For this reason, we may well separate the evolution of astronomy, the evolution of dynamic geology, and of physical geography, as well as the purely hypothetical evolution of chemistry, from the observed phenomena of the evolution of life. To regard cosmic evolution and organic evolution as identical or as phases of one process is to obscure facts by verbiage. There are essential elements in each not shared by the other, or which are at least not identical when measured in terms of human experience. It is not clear that any force whatever or any sequence of events in the evolution of life is homologous with any force or sequence in the evolution of stars and planets. The unity of forces may be a philosophical necessity. A philosophical necessity or corner in logic is unknown to science. We can recognize no logical necessity until we are in possession of all the facts. No ultimate fact is yet known to science. For reasons indicated above, the term evolution is not wholly acceptable as the name of a branch of science. The term bionomics is a better designation of the changing of organisms influenced through unchanging laws.